membrane is made up of lipids and proteins. Lipids include phospholipid, sterol, glycolipid. Phospholipid are of two types: glycerophospholipid and sphingolipid. As we know, glycerol is the three carbon structure. At the carbon 1 and carbon 2 we attach a fatty acid chain, and at carbon 3 we attach phosphate group. It becomes glycerophospholipid, and with that third carbon phosphate we attach a hydrogen atom. It becomes phosphatidic acid. Phosphatidic acid is the simplest glycerophospholipid, and it is a negative charge, negative one charge. And if we attach different alcohols, and the name change accordingly. If we attach ethanol amine, in the name becomes phosphatidyl ethanol amine, and it is present in the inner monolayer of the plasma membrane, means towards cytosol, and it is also present in the bacteria. And if we attach inositol, it becomes phosphatidyl inositol, and it is also present in the inner monolayer of the plasma membrane, and it is negatively charged, and we call it like PiP2. And if we attach serine, it becomes phosphatidyl serine, and it is negatively one charge, and it is also present in the inner monolayer of the plasma membrane, and it is absent in bacteria. If it flips toward the outer monolayer, it acts as a it-me signal or apoptosis. If phosphatidyl serine if present in the outer monolayer, it acts as a it-me signal. And if we attach choline as alcohol with the phosphate, it becomes phosphatidyl choline, and it is present on the outer monolayer of the plasma membrane toward exoplasmic phase, and it is also absent in the bacteria. So all these three alcohols are present inner monolayer of the plasma membrane, but phosphatidyl choline, it is present towards outer monolayer of the plasma membrane to the exoplasmic side. And the bond between the fatty acid and glycerol is ester bond. जो ग्लिसरॉल फैटी एसिड है वो ग्लिसरॉल से अटैच होती है एट कार्बन 1 and 2, then that bond is a ester bond. But in the case of plasmagenogen, which is found in archaebacteria, here is the ether bond, and ether bond is more stronger than ester bond, so ether bond cannot break easily. And the bond between polar head, means the bond between the phosphate and the alcohol is phosphodiester bond, means phosphodiester linkage is present between the phosphate at the carbon third and between the alcohol. And the basic backbone is here is a glycerol. And second type of this are sphingolipid. Sphingolipid are derived from sphingosine, which is the 18 carbon structure, and at the carbon 1 there is a OH group, and at carbon 2 there is amino group, and at the carbon number 3 again OH group is present. And if at carbon 1 we attach hydrogen atom, it becomes a ceramide, and ceramide is the structural parent of all the sphingolipid, ceramide. Okay, and it is synthesized in the endoplasmic reticulum, and sphingolipids are of two types, sphingomyelin and glycosphingolipid, which is also called glycolipid. स्फिंगोमाइलिन की बात करूँ, अगर कार्बन सीरामाइड के कार्बन 1 पे जो OH ग्रुप है उसके अगर हम अटैच करें फॉस्फोकोलिन, फॉस्फोइथेनॉल अमाइन, एंड बाय फॉस्फोडाइस्टर बॉन्ड ज्वाइंड बाय फॉस्फोडाइस्टर बॉन्ड, इट बिकम अ स्फिंगोमाइलिन. एंड स्फिंगोमाइलिन आर आल्सो प्रेजेंट इन द आउटर मोनोलेयर ऑफ द प्लाज्मा मेम्ब्रेन, एंड वी नो फॉस्फोटाइडल कोलिन and also present in the outer monolayer, and sphingomyelin is also present in the outer monolayer, and it has no net charge, and they have phosphate because phosphocholine and phosphoethanol amine are attached. So only sphingomyelin are the group of sphingolipid that contain phosphate. And next is glycosphingolipids, which are sugar. Actually glyco means sugar molecules are attached. So one or more sugar molecules are attached by a covalent bond to the OH group of the carbon 1 of the ceramide, and they are also present on the outer monolayer, and they does not contain phosphate. Okay, and it is of two type, neutral glycolipid and charged glycolipid. Neutral glycolipid include cerebroside and globoside, and charged it's include ganglioside. Neutral, we talk about cerebroside. इफ सीरामाइड, अगर सीरामाइड के स्ट्रक्चर पर फर्स्ट कार्बन पे जो OH ग्रुप होता है वहाँ पे हम मोनोसेक्राइड अटैच कर रहे हैं, इफ़ वी आर अटैचिंग मोनोसेक्राइड वाया ग्लाइकोसिडिक बॉन्ड, इट बिकम्स अ cerebroside. And if we attach glucose, it become glucosyl ceramide, and if we attach galactose, it becomes galactosyl ceramide. Okay, and if we talk about globoside, when di tri tetrasaccharides are attached, like if we attach glucose and galactose, it become lactosyl ceramide. Okay, here we are attaching like monosaccharide is attaching, we are attaching glucose, single sugar moiety. But if we are here, we are attaching two more, two like disaccharide, trisaccharide, tetrasaccharides, so it becomes globoside. If we are attaching glucose and galactose, it becomes lactosyl ceramide. And if in the case of charged glycolipid, when sialic acid is joined uh, at the uh, at the end term, we can say at the terminus place of the globoside के साथ अगर हम सियालिक एसिड अटैच कर दें, देन इट्स बिकम्स द गैंग्लियोसाइड एंड वी आर रिटन एज द GM3. G ganglioside M is like how many number of molecules are attached of sialic acid. Like if one sialic acid attached, we like mono. If two, we will like as the di. If we like three sialic acid, we are like tri. So GM means mono, and 3 here represent the electrophobic mobility of the molecule. Next we talk about sterols. Sterols are also structural which are present in the plasma membrane. In the case of animals, it is cholesterol, which is derived from the 5 carbon isoprene subunit, and it is synthesized in the liver hepatocyte cells of the in liver में भी कहाँ ER में endoplasmic reticulum. Hepatocytes, the cells of liver cells is called hepatocytes. In the case of plants, there is a brasinosterol, stigmasterol, and 2 for ethylsterol, and it is also present in two forms, alpha epimer and beta epimer in the case of plants. And in the case of fungi, it is a ergosterol. Okay, and sterols are absent in the bacteria. But recently we have studied that the primitive bacteria also contain sterol like molecules, which is called hoponoid, and example is bacteriohopenate tetrol, which is seen in the archaebacteria, which is synthesized in the archaebacteria. Majorly it is absent in bacteria. So it's just a quick revision on the lipids. Thank you.